Okay, so welcome everyone to this second workshop of our X-omics workshop series. Thanks for being here. So the program today is about linked data, including a practical example with genomics, and we are also sharing the link to the program in the chat. You can use the chat for commenting, discussing, or asking questions. So we don't have the Q&A feature today, and you can type questions at any time, but we'll answer them at the end and during the Q&A session. And you can also use the raise hand feature if you'd like to ask something in person. There will be 2 more workshops in this series. This week on Thursday, we'll learn about researchers' own experiences while integrating multiple types of omics data for 2 different studies. So we'll have Ayşe Demirkan, who will present her work, and there will also be plenty of time for discussion afterwards. And on Tuesday next week, you will have the opportunity to present and discuss your own multi-omics projects. If you haven't done yet, please fill in the separate form for this workshop. It's for both people who want to pitch their own project as well as participants. So we'll also share the link to that. And so this workshop series is part of the X-omics outreach and training activities. Please feel free to contact us if you'd like to share any comments on this workshop series, or if you have suggestions for future topics of X-omics workshops. I'd like to thank the entire workshop series organizing committee again, and also let me introduce your hosts today. So we have fewer people than on Thursday. My name is Anna Niehues. I'm a postdoc at Radboudumc Nijmegen, and with me is Jenny van Dongen. Maybe you can you wave your hand, from Vrije Universiteit Amsterdam. We have Jessie Smits and Daniela Castell from the X-omics project management team. As invited speaker, we have Roel Janssen from Center for Molecular Medicine at UMC Utrecht. And your chair today, and also the first speaker, is Jasmin Böhmer, who's a data steward at UMC Utrecht. And yeah, I'll just say enjoy the workshop, and then I'll give the floor to Jasmin.
Thank you very much, Anna. Let's share the screen. All right, also a warm welcome from my side. Again, my name is Jasmin Böhmer, and I'm here today joined by my colleague Roel Janssen. We both work at the Center for Molecular Medicine at the UMC Utrecht. I'm a data steward in Jeroen de Ridder's group, and Roel is a research analyst in the group of Edwin Kuppen. And we are more than pleased today to talk to you about linked data and practice, an RDF-based approach with SPARQLIing-genomics. This is the plan for our session today. First, I will introduce you into general terminology and concepts before I will hand over to Roel, who will introduce you to RDF and SPARQL and also demo SPARQLIing-genomics. Then we will have a quick intermission with an interactive quiz. And in the second part of today, we will talk about ontologies and FAIR data, and finish with the dedicated Q&A session and an open discussion. So let's start with the general introduction. In the next 15 minutes, I will introduce you to some basic concepts so that everyone is on the same page. I will familiarize you with RDF and SPARQL, and then, of course, highlight our SPARQLIing-genomics project. Let's start with the bigger picture. These days, we are facing a plethora of data formats, programming languages, tools, and technologies. And what we would like to achieve is to have some sort of interoperability across these data formats, platforms, but also research lines and research disciplines. In order to do so, we need to harmonize the data and metadata. In recent years, the interoperability movement got a renewed push by the FAIR data requirements, as they also have been incorporated by the funders these days and are a requirement in the recent research programs. Once we will have achieved some sort of interoperability, our data will be more reusable and more trustworthy. Let's start at the beginning. Here you can see a digital file. It's a spreadsheet and the format is XLS, which stands for Excel spreadsheet. When we have a closer look on the properties of this file, we can see what kind of Excel spreadsheet it is, how big the file size is, where it's currently stored, when it was created and modified, and things like this. These informations are regarded metadata. And in order to manage this file better among all the other files I have, I would like to add extra metadata. That means in addition to the original author, I also would like to know who's collaborating on this file. Maybe I already know what license I would like to attach. Even add a few more keywords so it's findable in my data jungle. And of course, set the access rights correctly so that only the right people can interact with this file, as well as the right machines. So now that we have looked at the overall file itself, it's time to look at the actual contents of this file. Turns out, in the spreadsheet is a table. A table consists of a header, columns, and rows. In this case, the header information contain information about sample, population, and other genotypical data. What would be great now if I could combine the data file contents with the information about the data file itself. That means I combine the general and extra metadata with specific metadata based on the data contents. That leads to information about the author and creator, access rights and keywords, but most specifically, it also includes the sample ID, the population ID, and the gender that is covered within this file. Combining all of these metadata parameters together is actually me creating a metadata standard. That means when I say that creator, creation date, gender, sample ID, data type, access condition, and so on are the set of parameters I want to cover, I sort of have created my own metadata standard. And now I would like to encourage my colleagues to capture the same metadata parameters so that we all have standardized metadata. The next step is to actually define what people should enter in these metadata parameters. I have chosen to say that in the creator field, I allow a free text field, which enables people to enter the names and titles of the creators and collaborators in any way or form. But for creation date, I have selected a specific syntax where I say we write the date in date.month.year. The other parameters are a mix of controlled and syntactical information. Now you might say, why do I need to control gender? It's 2020, isn't that by now a defined set of concepts? Here's an example of EBM, EMBL, EBI. They have covered how the term female is expressed in their data sets and infrastructures. And it's actually quite astonishing how diverse the way of expressing females is, isn't it? It all depends on the line of research that these people are doing. For example, when you research chickens, female chickens are called hens. But if you research specific insects, a Teletokkie is a female that reproduces in an asexual way. And of course, for humans, the phenotype female is most commonly used. Looking at this list, you can see that the semantic ambiguity is quite strong for the term female. And one way to decrease this ambiguity is to apply ontological terms. Here's an example of the Fair Genome Semantic Model. As you can see, they have decided to include the metadata element gender as well. They also have decided to use the ontological term from the SIO ontology to express gender. And that ontological term has two entities, one is male and one is female. and they added unknown to it. So we have 3 options in order to express gender in this semantic model. Now that we know we can use ontological terms to decrease semantic ambiguity and define ways of how to express real facts, it's time to look at how can we actually do this. Ontologies play a big part when you want to create linked data. And one way of creating linked data is to use RDF and SPARQL. RDF is also called the Resource Description Framework, and it expresses natural facts in triplets. You can see an example of a triplet. You can connect several triplets to each other and create a knowledge graph. On the other hand, you can see SPARQL. SPARQL is a protocol to query RDF content. With these queries, you can ask for raw values, create more triplets, and therefore construct information, or you ask the knowledge graph for true or false statements. Let's have a look at these triplets. This is a very basic and very easy knowledge graph. It consists of certain entities such as Alice, Bob, the Mona Lisa, and Leonardo da Vinci, and they all are related to each other in one way or another and also inherit certain parameters. So now let's ask some questions to this knowledge graph. When was Bob born? And the answer is Bob is born on 14th July 1990. Next question. Does Bob have a friend? Turns out Bob is a friend of Alice. And in our knowledge graph, Alice is a friend of Bob as well. It sort of implies that they are friends. That might not be the case in the reality, but in our knowledge graph, it's true. And now we can ask more complex questions to fully facilitate the power of this knowledge graph. Let's say we want to know, does Alice know a person that likes paintings of Leonardo da Vinci? The question actually has 3 parts. The first is, does Alice know a person? The second is a person that likes paintings. And the third is the paintings are of Leonardo da Vinci. Let's start. Does Alice know a person? Alice is a friend of Bob and Bob is a person. Check. Coincidentally, Bob is interested in the Mona Lisa and we know the Mona Lisa is a painting. And of course, we know the Mona Lisa is a painting by Leonardo da Vinci. So the answer to this specific query would be yes. Alice knows a person that likes paintings of Leonardo da Vinci. Looking more closely at these triplets. RDF triplets have a specific syntax. They consist of 3 parts: a subject, a predicate, and an object. The subject always points to the object, and in this example, Bob is the subject, is interested in is the predicate, and the Mona Lisa is the object. Now we look at a real-life example of a small knowledge graph based on RDF triplets. Here we can see in the center a specific protein. It has a short name and a full name and a synonym and a sequence and cross-references and so on. As you can see, in these objects, there are quoted elements, terms. And as we know now, these terms can be free text fields or controlled terms or even ontological terms. Moving on to the current omics landscape. We are, of course, faced by a wide variety of programming languages, file formats, and standards. For example, there are BAM files for genomics, which are binary alignment map files. Then we have NMR files for metabolomics, which are nuclear magnetic resonance spectroscopy data. And for proteomics, we have mass spec XML. Some programming languages are used in all the omicses, like Python and R. Other programming languages are very specific. And lastly, of course, there are standards, like ISO, but also metadata standards and ontological standards. So we face quite a big challenge to harmonize and interoperate between all of these different omicses. And now I would like to introduce you to one approach of improving the interoperability for genomics data, which is our SPARQLIing-genomics project. Roel and I worked on this all last year. We had a 1-year pilot project where we took the variant call format from genomics, which is a standardized file format, transformed it into RDF triplets, while also creating an appropriate RDF triple store. And we designed a web interface, which is powered by SPARQL to interact with these triplets. Roel created a bunch of tools that enable to take certain elements of files and transform it into these triplets. So we have VCF to RDF, table to RDF, XML to RDF, JSON to RDF, and so on. These tools enable us to take file-specific information, but also information from within these files, and transform it into RDF triplets. The next step was to enrich these triplets with a variety of ontological terms and metadata standards. We used quite a few, among others, Dublin Core Metadata Standard, the gene ontology, and the data use ontology. We have been quite successful last year and can proudly share with you now a fully functional SPARQLIing-genomics portal, which you can find on sparqling-genomics.org, as well as an exhaustive manual that is freely available for your consumption. And without further ado, I will hand over now to Roel, who will introduce you and demo SPARQLIing-genomics and RDF triples.
Thank you, Jasmin, for the introduction. I would like to let you go through the process of going from a spreadsheet to a SPARQL query. And I'd like to show you in the meantime how SPARQLIing-genomics helps you finding the data structure of your spreadsheet. Now, before we go into the demo, I'd like to go back to to a question from the previous workshop, where the participants found that the skills needed for big data and multi-omics data integration, the top 2 skills would be knowledge on data structure and programming skills. Now, I think that with RDF and with some visualization tools that I will show you shortly, we can address the first 2, so knowledge on data structure, and we can reduce the need for programming skills by using SPARQL to solve problems instead of encoding it in more generic programming languages. So without further ado, let's go to the demo. So for the demo, I'm going to download a spreadsheet from the 1000 Genomes project, which you can find on their website. Now, so we've seen the format of a spreadsheet probably often enough to understand it. There are columns and rows. The first step is to get rid of the Excel format and put it in a format that's well, more open for other programs. So I'll save it as a comma-separated values file. Well, technically, I used a tab now instead of a comma as a separator, but that doesn't matter. So we now have this this file, and that's still not RDF. So I'm now going to use one of the tools from SPARQLIing-genomics called table to RDF to convert this comma-separated values file into a RDF file in the n-triples format. And then I'll show you what this RDF actually looks like. We won't look at it too long, but just to get an idea of what it is and how it looks like. So this is the RDF file. It looks uglier than the table, doesn't it? But the file format is basically every line contains 3 elements. It contains a subject, a predicate, and an object. And I hope you can see the blue highlighting on my screen. So every line contains the same 3: subject, predicate, object. And by reusing the same subject, as we see with this description for a sample column, we can add more properties to a single subject. So this representation is text-based and not great, at least not for everybody. So let me show you our web interface that also shows you the structure of the RDF file, but then, well, hopefully a little bit better. So first we have to create a project and give ourselves a graph, because we want to store this data in an RDF store before we can do anything else. So the next step would be importing. Now this is just implementation details, so just bear with me for a moment. So the web interface tries to be very honest and transparent, and therefore it sometimes gives you a large command. But this command is completely independent of SPARQLIing-genomics, so you can just use this for other tools as well, and you can find out what exactly you have to do as a programmer to do the same thing. So I'll copy this command, and let me go back to the terminal. And now I can paste that command and give it the n-triples file that we generated with table to RDF. And because we asked for an XML response, it'll respond in XML and say this file has been imported. Great. So now that we got all these details out of the way, let's start to look at the structure of the RDF. So we'll have one connection. This is the graph we created. And now the structure tool gives us 3 types of information that we have. The first type is the metadata, and currently it stores the checksum of the CSV file, the file name, and some information about how it was converted. So basically it states it's converted by table to RDF. And the second piece of information is the information about the columns. So if you'd like to go back from RDF to a table, this information allows you to have the first column back as the first column in your regenerated CSV file. But then the most important piece of information is what properties can each row in the RDF be queried on. So these properties are the properties that we can use. And what we usually do in our lab is we do a split window view of both the exploratory and our query. Because now we're ready to build our first query. So let me remove the parts that we don't need. So, okay. So we're going to retrieve some information. And the first thing we have to think about, the SPARQL query will result in a table. So the first thing we have to do is think of which table columns we would like to see. So let me write down 3 table columns we'd like to see for the sake of this example. So let's take sample, population, and gender, because we used gender in the previous examples. Next piece of information we have to give the query is which graph would we like to search the information for. So let's use our graph. And now the hardest part of the query, and actually the most fun part, of course, is how are we going to retrieve these three columns of information. Now, we have a row for which we have these columns. So let me type this out. So we specify that there must be a triple for which we only know the predicate is this column prefix sample, which should be in the data because we found it in the exploratory. Now this same row, so we use the same keyword, should also contain a column or value for the column population. All right, and then the same thing for gender. Now let me format that a bit. So each statement consists of a triplet in which if we start the variable with a question mark, we leave it open for the query to fill in the details for us. So we're now going to search for rows which contain these three predicates. And these predicates are linked to our output columns. And for the sake of the example, I'm going to limit this query to 10 results. So the results is a table with our three columns. And for each variable in our query, the solution is given. And we only ask for 10 entries, so we get 10 entries. Now, this is something we could have done with the spreadsheet as well. And the next example we can also probably do with the spreadsheet, but let me just go on and show what other things you can do with SPARQL. So let's say if we want to count all the males in a certain population, then we'll specify, we use the count function on the gender column. But now it's males and females, so we'll have to introduce a filter as well to say that the gender must be male. Now, this tiny thing is an implementation detail. Don't get too worried about it. So if you execute this query, we'll get a listing of each population with the number of males in that population. Now, this short code for the population tells you something, but maybe we could also change this to a description of the population. Okay, so that's already a bit better. Now we can order it. By the number of males in population, so that we get the population with the most males in it, male samples in it, as the top entries. So now we see that there are two separate entries for Nigeria. So if we want to count the total number of male samples from Nigeria, we could introduce another rule to say, so the population variable, which links back to the population description, must contain the word Nigeria. And that will give us the sum of the top two entries. Now, to be correct, we should state this in our variable. Which, well, so this is what SPARQL looks like. I hope to have shown you an overview of a way to use SPARQLIng-genomics to get through the structure of your RDF. You can use, so we use table to RDF in this example, but we also made tools for XML to RDF, for JSON, for VCF and BAM files. So tables are pretty transparent, so you could use a spreadsheet for that. XML formats contain more hierarchies, so you'll probably need a better tool to explore the structure there, and I think that's where the exploratory can help you out. So this is probably a lot of information to absorb, so let's have some fun with a quiz before we go to the last part of this workshop, which is actually verifying these RDF triplets.
Great, thank you, Roel. I will share my screen again. And now we will start the quiz. Okay, please use your laptop or a tablet or another device and go to menti.com, entering this code. Once you're there, please also click the little heart symbol to indicate that you're there, and I will wait a few minutes for you to join. It's a quiz tool, it's anonymous, it's non-competitive, so please join if you would like to interact in our quiz time with us. I will wait 1 more minute and then we will start. All right, we have 25 people that want to play. Great. Let's start with the first question. We would like to know, no, we are waiting for players, and now we will start. Okay. Please don't answer fast, there are no points. This is non-competitive today. So we would like to know, how do you currently store your metadata? Are you using a spreadsheet, or are you old school and using pen and paper, analog? Or are you all doing this in your head? Maybe some of you are actually using RDF or a relational database. Please vote for your choice. Is it not working? Or we see the answers when the timer is done, I guess. Ha! Ooh, interesting choice. Spreadsheet mainly, 2 relational databases. Nice. I think this is a good discussion point for our Q&A later. No one's using RDF now. All right. Next question. What is a teletalkie? Please write down the answer if you can remember what it is. I've mentioned it briefly in my presentation. All right, 5 seconds left. Nice, female bug. Yes, yes, yes. Good. We allowed ourselves a little joke because this Wikidata identifier is the correct answer with regards to linked data. But of course, female is correct, insect is correct. And if you wrote down in any form that it's asexual reproduction, that's also correct. Unfortunately, Mentimeter is really strict with the way I typed in answers and checking your answers. Obviously, on a conceptual level, a lot of you were correct as well. Well done. Next question. What does RDF stand for? And we offer you a bunch of options. Is it Resource Definition Framework, or R Database Format, or Resource Description Framework, or Rare Data File, or lastly, Roaring Dinosaur Fish? Please make a choice. You have 25 seconds left. All right, let's see what you went for. Nice. 20 people went for Resource Description Framework. That's correct. Of course, we sneakily used wrong terms that are pretty close to the original meaning of that abbreviation. And last question. What does the SPARQLIing-genomics logo looks like? Is it a pear? Is it a turtle? Or is it an owl? I hope you paid attention to our cute little logo as well. You have 30 seconds left to make a choice. Please select one of these three. Ten more seconds. Nice. Well done. It was actually an owl. Turtle and owl are ontology-related terms as well, and sadly, no pear today. Thank you very much for interacting in this short intermission quiz with us. And now we will move over to the second half of our workshop, and we definitely need to talk about ontologies now, and I will hand over to Roel.
All right, so I hope the quiz was fun. So we need to talk about data standards. Vocabularies or ontologies. Let's talk about way to name things and to be unambiguous about what we say. So let's go back to the ambiguity example of AMBL EBI. There are many ways to say female, and some are more specific than others. So hen and talitogi was more specific than F or female. Now, some terms overlap in their specificity, at least I would guess. For example, F email and female written the usual way, I think just mean the same thing, but who knows? So you might encounter in your data that you have many of these terms for which you think the meaning is the same. So if we would create RDF triples of these terms, then we would find a subject, S1, S2, or S3, with the gender predicate, and then a specific term for specifying the gender, in this case female. Now, if we would like to unify our data in a way that we can later more easily get an overview of all the female samples in our database, then we could add for each specific entry of female an extra triplet saying so this term that we see in the middle is equivalent to, and then one unified term for female. And if we do that, we can build a SPARQL query that asks for subjects for which there is a gender predicate, and then we leave the gender open as a variable, which we then reuse and say the gender must be equivalent to female, and that will give us all three subjects back. Now, if we would have just not used this variable gender here, but specified female here, we would only get a single entry back, namely S1. So that's one way to remove the multiple terms saying the same thing in your data by just unifying, by adding some meaning to it, like you just meant the same thing or meant the equivalent term. But that's not so great for a complete solution if you'd like to share data with other people. And this is where the Fair Genomes project does a really good job because they defined the terms in community set standards. So they used the SIO terms for gender. So that's a really good idea to publish your data definition by using community defined ontologies. And then just publish this list of terms that you're going to use on your source code page or on your website. So in this case, you use the SIO terms, but we didn't use that in our spreadsheet, obviously. But we can use an insert query from SPARQL to use these SIO terms. And the query reads as following. So insert into a graph of our choosing this triplet. So for any gender that comes through, assign the SIO predicate for gender and then the SIO object for female. And do that for every gender that is equivalent to female. And now for this term, for our gender terms, we have made ourselves compatible with the data standard set by the Fair Genomes project. It could very well be that there's another project that set different standards that perhaps used the phenotype and trait ontology to define gender. So adopting one single standard might not be the most compatible way we can be, but transforming data from one form into the other makes us very flexible in becoming interoperable with a very specific project, and then we can just flip from one project to the other by inserting a couple of more triples to our database. So by using the resource description framework, we might be able to reduce the format specificity, and Genomics has their BAM and BCF files. We might be able to extract knowledge from that and put them in a resource description framework, use terms that are also used in other fields, and therefore we could unify our data into a linked structure, which we can then use. So we went from a spreadsheet and we used the terms for we used the columns of the spreadsheet as terms in our RDF graph. We've seen a SPARQL insert query from which you can go from one local data standard to a more general data standard set by somebody else. And this is a picture of how that would look. So if you go from your spreadsheet and then project the data in a way that's compatible with the Fair Genomes project, then that's one projection of your data. But then again, if there's another organization or project that has a different data standard, then you just make another projection of your data. So just another insert query to augment your data to allow it to be interoperable with the various projects there are. So that brings us to the last two slides, and I'd like to give the word back to Jasmin for this.
Thank you, Roel. All right, so Roel has shown you how to create fair data with ontological terms. And now I would like to tell you what you can do if you do not have the resources like RDF and SPARQL at hand currently, but still would like to verify your data a bit better. That is my personal opinion and has not yet been proven successful. Nevertheless, I think it's a good suggestion, and we can of course discuss it later in the Q&A. The first step is to select a standardized and certified data archive or repository, such as 4TU Center for Research Data or Zenodo. These specific archives will ask you to submit metadata in Dublin Core already, which is a metadata standard, and they will offer you keyword fields, and in the case of Zenodo also these subject fields, to enter more specific domain-relevant metadata terms. So what you can do is, with the aid of Zenodo, is to facilitate the subject chapter in their upload form. That means you can look up ontological terms in an ontology of your domain and paste the exact wording into the subject term field shown here, and also enter the ontology ID for that specific term in the other field. And of course, you can add more metadata. If the subject feature is not available, but keyword fields with free text fields to enter, like in most data archives and repository, I would like to suggest to you to go to ontology of your choice, of your domain, look up a specific word that describes the keyword you wanted to use in a very harmonized and standardized way, and copy and paste that exact wording into the free text keyword field. Both approaches will improve the harmonization and interoperability of your data set with other machines and humans, as you will use the standardized and harmonized terminology. Overall, of course, when you use standardized and certified archives, you will get a DOI, metadata, licenses, and specific file formats, which will make your data more fair. And with this little workaround, I think we can even increase the fairness for the most relevant domain-specific terms. I would like to point you to a really good resource called fairsharing.org. It has been created a few years ago and is attempting to be the most up-to-date and unifying platform where you can find standards, databases, policies, and so on across all research domains. Sometimes when you look up ontologies or metadata standards from specific research lines or domains, you might enter a website that give you a 404 error message, or you simply see that this standard has not been maintained for a couple of years. Fairsharing.org is maintained by the publishers of each standard, and you can be sure that all of these standards are up to date, and you can find documentation and implementation help how to apply it. I really hope that you will find the necessary information for your specific line of research. And if you encounter that your specific research domain has not yet developed standards, I encourage you to consider contributing to this and maybe get in touch with your colleagues and start an initiative to create a standard for your research domain. to summarize our presentation part of this session. We can create a knowledge graph of diverse omics data using RDF. And ontologies help us unifying terminology across research domains. We can transform a spreadsheet into ontologically defined data. And of course, flexible data management requires easy data transformations, which can be done with SPARQL. And lastly, SPARQLIng-genomics is a starting point for interoperable omics data, and we highly encourage our colleagues across all the omics to consider RDF and SPARQL to try to achieve interoperability for your omics type. And without further ado, we are now segueing into the Q&A part of the session and the open discussion. Please use the chat to ask your questions or the raise hand function, and then we will enable you to talk. Thank you very much for your attention. All right, the first question is from Yannick, and I will address it to Roel. Yannick is asking, triples were mentioned in the first presentation, but I didn't hear anything about quads. Some people claim storing the provenance metadata is vital for working with big data and semantic stores. Could you explain to a layman why you opted for triplets over quads?
Yes, I will try. So quads are, from a technical point of view, basically a triplet inserted into a specific graph. So in the demo I gave, we actually used quads once we inserted it into a graph. Now, you can use this graph name to denote the origin of where this data came from, so that the graph names are similar if different people import their data in different systems, in their own systems, then you can still know where this data came from. So that's one way to do a bit of adding a bit of provenance information to your graphs. There's also other mechanisms of adding provenance. For example, the MD5 checksumming is used to not only verify data, but also make sure that you're using a very specific version of a data set. So quads can be used for adding some provenance information. I don't think you can encode all the information in it, but it's definitely a good way. So yeah, do use quads if you think you need to unify the graph name. In our example, we use triplets, but yeah, I agree, quads are just as good or better.
Great, thank you, Roel. There's a next one from Hank. Is it possible to capture the strength of the connections in predicates, or is this pure logic?
So does the question ask about how you could infer data, or sorry, I might not understand the question completely.
No worries, Hank. You are free to speak up if you want to. Otherwise, please type again in the chat what you exactly want to know. Oh, okay. Yeah, please use the chat since your microphone is not working. We will wait. The slides of today's session will be shared. We have actually already published them. I will paste the link in the chat in a few moments. And of course, the recording of the session will also be made available. Okay, Roel, can you read his question out loud and answer it?
Yes, so the question is, sometimes knowledge is not 100% sure. For instance, if you have a p-value for a relation, can this be expressed and how? So the RDF framework is pretty much just what you see is what you get. So it's pure logic, but you could add the p-value as a property of a subject. You can't do like dynamic types, like changing the p-value in the triple sort, because once you write it as a triple, then that's that. You could use the p-value as a predicate, but yeah, I don't know what. So you should always consider how will you query your data later on, and pretty much form your RDF towards what's easy to query in the end. So I don't think RDF is completely flexible as that, but maybe we should look at some examples after the session to get to the bottom of this.
Sounds good. Thank you, Roel. Coming back to the Mentimeter insight that we gained earlier. So we asked, how do you currently store your metadata? And the overwhelming majority said in spreadsheets, and 2 of them said relational database. So I'm still a bit surprised, and I would appreciate if a few people could speak up that have participated in this survey, or in general want to say something, because I'm really curious if you are applying metadata standards in your spreadsheets, or what is the structure of the metadata within these, let's assume, tables. You can use the raise hand function if you like, or type in the chat. All right, it seems that no one has the urge to delve into that deeper. That is fine, though. All right, are there any further questions, remarks, or discussion points? Then please write them in the chat now, or raise your hand so we can enable you to talk.
Yeah, I have a question myself.
Mm-hmm.
So I've asked the question to several people. So how do I find the most appropriate ontology for my data? And that seems to be a rather difficult question to answer. But after your talk, I have the feeling it's not so important which one I use, but that I start using one. Would you agree with that?
That is correct. In my experience with Roel together last year, since he is obviously the expert on RDF and ontologies and so on, I realized that what I like to call ontology cherry picking is totally legit. So if you have multiple ontologies that you can use, and you use the most appropriate terms for your line of research from each one of them, that is possible and totally fine. And yes, you are right. Baby steps are also totally fine. If there is an ontology that is available and only one term fits, then use that one term. It will be a slow process for us to achieve interoperability, but this is a way how everyone can contribute in harmonizing data. All right, another question from Hank in the chat. How do you get people to use ontologies to describe their research? Maybe Roel, do you want to answer?
Yeah, that's indeed a very, very good question. From my experience, people just don't. So the best way to make people use ontologies is by large projects to provide their data in a form that uses the ontologies already, so that the easiest way to get data to be compatible with the biggest data sets to use the same ontology terms. It's a bit of a might be perhaps a strange solution, but I think that's just the way it works. If you don't have a lot of data, then you won't have any influence in what ontology is actually going to be used. But if large data providers do use an ontology, and that's why I like the Fair Genomes projects, for example, because they specified already what they're going to use, then we might have a shot at everybody making themselves compatible with this one big data set because they want to compare their data with it, and therefore use the ontology terms used by the bigger project.
And of course, bringing the X-omics initiative into the discussion. X-omics just has started and is a very long-term endeavor. And of course, we are all very hard working on harmonizing data across the omics. The Fair Genomes project will feed into that for the genomic data, and hopefully there will be other projects for proteomics, metabolomics, and so on. Of course, currently you are receiving a workshop from the Data Integration and Data Stewardship pillar, but this is of course what this workshop series is all about. How can we enable interoperability across omics data in the future? Yes, and Henk remarks that there are some tools for proteomics, which are not using RDF currently in his opinion, but we will have a look. All right, are there any other remarks, discussion points, feedback? Okay, I cannot see anything, and therefore I would like to thank you very much for your attention and hand over to Anna to close the session.
Okay, thank you, Jasmin and Roel. Thanks for your talks. It's very much appreciated. And thanks everyone for attending. If you have any questions, you can also contact the X-omics help desk. I'm sharing the contact information in the chat. So feel free to contact that. And then I hope to see you again on Thursday. Closing the session now. Okay, thanks. Bye.